it gives me great pleasure to introduce Dr. Sue O'Shea, who is the director of the Stem Cell Core Laboratory here at the University of Michigan. A number of years ago, when stem cells were emerging as a focus of research in medicine, we began thinking about it here at the University of Michigan, and it was clear. I remember I told you I ran a laboratory at Hopkins, and it was clear to me 8 years ago that it was not going to be a good idea for me to attempt to run a laboratory here, rather to work with the individuals that do this work. And so Dr. O'Shea, as the director of the Stem Cell Core Laboratory, was singularly the best person to go to. She has trained most of the individuals here at the university that are working on stem cells. The Stem Cell Core hosts training for investigators, junior and senior, to get them up to speed with the work with stem cells. And so over the past couple of years, we have been working tirelessly to set up the bipolar stem cells, and we're so excited to have you come and speak with us here about the work that you've been doing here with us. Thank you.
Okay.
So this advances.
I think I'll probably do this.
Okay.
Will that work?
Yep.
Okay, great.
Now, you can either speak into these, or we can use...
I think I'm going to try to use the microphone so I don't walk all over the place, but
Okay.
there's also stuff. Okay, thanks very much, Melvin. Thanks to all you folks who braved the beautiful Michigan fall weather to come here today. Since I'm the first speaker today, I wanted to just begin by giving you a little bit about background of embryonic stem cells, pluripotent stem cells, induced pluripotent stem cells. I want to give you a little bit of an idea about where they come from, how we get these stem cells, and what they can do. Good. So we need a little bit of vocabulary before we can talk about stem cell biology, because these cells behave a little bit differently than most kinds of cells in the body. So in order to be considered a cell must absolutely have a characteristic that we call self-renewal. So with every cell division, a stem cell, unlike most types of cells in the body, gives rise to committed cells that are going to differentiate into other lineages, but it uniquely replaces itself. It self-renews. So at least in theory, the stem cell population in the body or in a tissue culture dish should never be depleted. This is a unique feature of stem cells. Another characteristic that a stem cell must have is that it must be multipotent. So if a muscle cell divides, it gives rise to another muscle cell, or 2 muscle cells that differentiate into 4 muscle cells. What's unique about a stem cell is it doesn't form just one cell type, but it has the capacity to form multiple differentiated derivatives. So for example, a neural stem cell in our brains doesn't just form new neurons, it also has the ability to form glial cells. So multiple choices, multiple chances to differentiate into various lineages really characterize a stem cell. There are many, many different types of stem cells. I'm going to talk to you a little bit about 3. The first is adult or tissue stem cells, and it's turning out that as we do more and more research, every organ system, every tissue in the body has a resident population of stem cells. We can't always identify them prospectively. We know they're there. They have a limited, typically, ability to divide in the organ. They replace themselves with every cell division. And they also form typically the kind of tissue that they come from. So a neural stem cell, when it differentiates, doesn't give rise to bone. Unless we do something exotic to it, it gives rise to neurons and glial cells. And bone-forming stem cells don't typically form neurons, but only bone. So that's a bit of a limitation with these stem cells. But what's tremendously important is that if we could figure out how to take tissue stem cells, convince them to divide, multiply, and then migrate to an area of interest in the body, those cells would be immediately matched to the person who needed the stem cells. There'd be no concern with having to implant stem cells. They'd be immediately matched to the donor. Okay? So if a person has a myocardial infarction, why isn't that we can't motivate the heart stem cells to replace those lost cells? How can we do that? Okay, you can see that there would be tremendous advantages to tissue engineering if we could just understand this behavior. Embryonic stem cells, on the other hand, are derived from the early embryo after fertilization, but before it's even implanted into the uterine wall. So these very primitive cells have the ability to form all the cells and tissues of the body. Okay, so that's a tremendous advantage, these embryo-derived stem cells. They have an unlimited ability to divide in tissue culture. So if we put these cells into a tissue culture dish, they divide really rapidly. That's great for us because it gives us a lot of starting material to study, but that's really bad if we put one of those cells into the brain or into the heart and it starts to divide. There's a concern that you might form a tumor. Okay, so the point really is that every stem cell type has limitations and advantages. And there's no right stem cell population to use. We need to do research and learn about the characteristics of all the various stem cell types. Induced pluripotent stem cells are definitely the new kid on the block. They're kind of an amalgam between what we know from embryonic stem cells and adult tissue cells, not adult stem cells, but tissue cells. So what scientists have done for over the last 6 years or so is to take genes that are expressed in the embryo and in embryonic stem cells, express those in adult cells, perhaps skin cells. And they're able to turn back the clock. They're able to reprogram these cells to behave as though they're embryonic cells so that we can study differentiation of cells that were taken from an adult for the very first time. Okay? I'm not going to talk to you very much about adult or tissue stem cells. I wanted to give you a couple of what I think are very interesting examples about these cells. The first is fat stem cells, and I'll be the first to line up to donate if there's a clinical trial, or thanks, see me afterwards. Who would have thought that fat was a stem cell reservoir? These cells don't transdifferentiate into neurons or bone-forming cells, but they do have the capability of forming muscle and cartilage, tendon kind of cells. Another really interesting reservoir, I think, is baby teeth. Who would ever have thought that shed baby teeth or shed wisdom teeth are a reservoir for bone-forming cells? So maybe we should be banking our kids' shed baby teeth, sort of the stem cell tooth fairy, if you will. I think then what most of us think about stem cell biology, we think about what we could do with pluripotent stem cells to replace cells that are lost or damaged. These cells could be lost and form a birth defect. They could be lost following an injury, a disease process, or even the normal aging process. So if following chemotherapy we need to replace the bone marrow, wouldn't it be great if we could take pluripotent cells and coax them to form cells that are typical of bone marrow for transplantation? We think a lot about taking pluripotent stem cells and differentiating them into nerve cells to replace cells that have been lost in Parkinson's or in Alzheimer's disease. I can never help myself. This slide came from the NIH website, and they can't spell Alzheimer's disease, which I think tells you quite a lot about our NIH these days. Heart muscle. I mean, surely we should be able to figure out how to differentiate cardiac muscle. Pancreatic islet cells. There's tremendous interest in making beta cells that might secrete insulin to treat diabetes from pluripotent stem cells. And I think that there are a number of different ways that we can also use stem cells to inform us. First of all, we can use, if we could take a whole plate, a tissue culture dish full of, for example, neurons or heart cells, why not use those as the starting platform to test new drugs, to do toxicity testing instead of using rats and mice and non-human primates? Isn't this a great source of human material that we could use as a test bed? Another thing I think we can do really efficiently with these pluripotent stem cells is to study differentiation. And this is what my lab does. I'm really very interested in studying the early steps of human embryology. And this isn't something we can obviously do ethically, legally, or in a tissue culture dish. So for the very first time, we have a source of normal cells, the embryonic stem cell or the induced pluripotent stem cell, that we can study the stepwise differentiation of a pluripotent cell into, for example, a neuronal cell. For the very first time, we can do this in a dish. And I think that our research group is really focused on taking stem cells from individuals who carry disease, genetic disease, both adults as well as embryos, so that we can model disease in a petri dish and really understand the stepwise differentiation of these cells. So I think that stem cell biology offers hope for a lot of people, and particularly people who suffer from chronic disease. At the same time, there's been a tremendous amount of hype. Turns out that everybody from Liz Taylor to Barack Obama has had some kind of stem cell transplant, and it's up to us now to really dial back the hype and really focus on what these cells are really capable of doing. I want to tell you a little bit about embryonic stem cells because basically they're the gold standard, the standard against which we compare every other stem cell line. So embryonic stem cells come to a laboratory via in vitro fertility clinics. The cells are typically 1 to 2 days post-fertilization when they come into a laboratory. The embryo continues to develop. You can see here that this is an early blastocyst stage embryo. The kind of light green cells around the outside of the embryo are destined to form the placenta. Their trophoblast cells are going to be responsible for implantation of the embryo into the uterine wall. And there's a cluster of about 100 to 200 cells that are in kind of flesh tone. These are the inner cell mass, and these are the cells that are going to give rise to every cell in the embryo itself. They're pluripotent. We put them into a tissue culture dish and allow them to grow, and they divide very rapidly, forming a stem cell line. So one embryo, one embryonic stem cell line, okay? And in the spirit of ethics and policy and regulation today, we're allowed to make new embryonic stem cell lines from early embryos until the 14th day of differentiation. The idea is that lineages, particularly brain cells, start to form at about this time. And I think the concern by policymakers and bioethicists has been that we don't really understand that this embryo doesn't or does feel pain. And so we want to avoid that by all means. These are what human embryonic stem cells look like through a microscope in our lab over at the BSRB. These are clusters of thousands of human embryonic stem cells from a single cell line that are growing in tissue culture. I wanted to tell you just a little bit about, let me see if I can figure this out, about what we can do with these stem cells. So obviously we'd like to cause them to differentiate so that we can implant them following injury or disease or aging. This is an example where we've differentiated human embryonic stem cells into cardiac cells. You can see they beat. In a minute we'll have a higher magnification view, and you can see that there are actually blood cells that course through endothelial line channels, forming vessels. Obviously, so you can see maybe you guys in the better seats than I have, there's some channels and there are red blood cells that course through those channels like blood vessels. So obviously this isn't an organized heart. Nobody's talking about doing heart transplants from stem cells, but you can imagine that this would be a great patch if you had a myocardial infarction, a heart attack, and you needed some replacement cells. You can see that you might use this for a baby who's born with a hole in its heart, a ventricular septal defect. This might be a great source of cells in those conditions. So again, with the ethics, bioethics flair today, I wanted to mention another study. This was a study that scientists did about 6 or 7 years ago now. They took human embryonic stem cells and caused them to express this green dye so they could trace them. They then took the human embryo cells and put them into chicken embryos. This was a real legitimate experiment. We need to know that the cells will continue to live. We didn't know that. We want to see if they'll interact with the chicken cells, make connections, send out processes. Would this be possible to engineer the spinal cord after, for example, an injury? Okay, well that was fine. They labeled the cells with the green dye. They put them back into chicken embryos. And surprisingly, I think to many of us at that stage, the cells did integrate. They lived, they formed connections, they sent out axonal processes. But this really got the attention for the first time of the bioethics committee. And bioethicists started asking questions like, how many of these human cells can you put into a brain before the chicken embryo starts to have human thoughts, before the chicken starts to have human feelings? Okay, what would happen if one of these human cells fused with a sperm cell or an oocyte? Would it be possible to transmit human genetic material through the chicken germline? Okay, it seems ridiculous that we might have a chick-human chimera, but this was the first time that we started thinking about how we ought to regulate this field. So a panel from the National Academy of Sciences got together and put in place some regulations. And one of the regulations we live with is that we can't breed these animals. So there's no concern that the genetic material will be passed on. We need to transplant cells to model organisms. But if we don't breed them, then there's no real concern about transmitting the genetic material. I want to talk to you a little bit more about embryonic stem cells and what they can tell us. I told you they're the gold standard by which we evaluate every other stem cell type. I hope that I convinced you that we can learn about human development by studying these cells. They've informed cancer cell biology. There's a whole theory, a whole bit of research about cancer stem cells now that is informed by human embryonic stem cells. But there's a big interest in understanding what the factors are that keep these cells undifferentiated and pluripotent. There's interest in rejuvenating tissues. And a few years ago, a couple of scientists, particularly Dr. Yamanaka in Japan, said, what would happen if we took the genes from embryonic stem cells and put them into adult cells, like skin cells? What would happen? And what they found is that they could push the clock backwards, and suddenly these adult tissues would start to behave as though they came from the early embryo. And these two guys, John Gurdon in Britain and Dr. Yamanaka in Japan, just in October of this year, shared the Nobel Prize for Medicine for these discoveries about stem cell biology. So basically the goal was to take an adult cell, like a cell from your skin, express these pluripotency genes that we know are expressed in embryonic stem cells and in early embryos. We force the fibroblasts to express them, and then they reprogram themselves, essentially setting the clock backwards in time so that they behave like embryo-derived cells or induced pluripotent stem cells. And these cells are really very important because they came from a person who might need a stem cell transplant, personalized medicine. They also have the ability to differentiate into the many lineages of cells in the body. This is what it looks like as we reprogram cells under a microscope. So these are dermal fibroblasts that someone has donated to us, a skin sample that we then take fibroblasts from. We introduce 4 reprogramming factors into these fibroblasts. They change their behavior. They go from this flattened sort of morphology to forming colonies or clusters of cells that resemble they're very similar to the morphology of embryonic stem cells. And of course, our goal is to take these pluripotent cells and differentiate them into neurons. And this is often referred to as brain in a dish. So why are induced pluripotent stem cells important to study bipolar disorder? And I think this group particularly understands that bipolar disorder is an unfortunately common, it's a very complex condition. We know that it's highly hereditary, and yet we don't have a solid understanding of the genetic basis for bipolar. And at least in my mind, there are a number of reasons that we haven't made a little more progress in understanding its biology. The first of these is we don't really have a good cell-based model. So people have taken brains at autopsy from people who have been diagnosed with bipolar disorder. They've put those cells, the glia and neurons, into tissue culture, and this can be very effective. But unfortunately for bipolar, there isn't a nucleus that's called the bipolar nucleus. We're not entirely sure which neurons and glial cells we ought to be sampling. I think the other problem is at the end of the lifespan, when we're taking cells from cadaveric brain, there's a real concern that you're looking at the end point of what's essentially a degenerative process. I don't think that anyone would argue that at the time of diagnosis with bipolar disorder, that this is when the disease became obvious. Surely and presumably there are a series of changes in the brain that lead up to this diagnosis. So by dialing back the cell from the adult phenotype to an embryo-like phenotype, we can then look at the differentiation of those embryonic cells as they form neurons and as they form glial cells. And this allows us for the very first time to really see when, during the differentiation of a neuron, things start to go awry. Okay? So hopefully we can target those steps in differentiation. It will tell us what processes are going on, and hopefully that'll lead to a new way of thinking about the development of this disorder that will eventually improve our understanding of the disorder and suggest new therapeutic approaches. I think that there's another really good reason to study induced pluripotent stem cells here at the University of Michigan, and that's because all of you have developed this amazing Prechter database that has so much information about individuals, their families, their genetic makeup, their heredity, their neurophysiology, and hopefully stem cells will form one component that helps us understand this disorder. So our goal was really to develop a cell-based model so that we could study bipolar disorder. And what we've done is obtain skin samples from patients both diagnosed and not diagnosed with bipolar. We've transformed those to induced pluripotent stem cells, and we're now differentiating them into neurons so that we can study this stepwise differentiation. So this is basically the stepwise procedure we do. We get individuals to donate skin samples. We take from those fibroblasts, and you can see that fibroblasts are pretty uninteresting little cells. They're branched. There's a lot of space between fibroblasts, and when we introduce the reprogramming genes that we know from embryonic stem cells, they start to change their behavior. They aggregate. They eventually become an epithelium-like cell. You can see that here's a colony after about 14 days after transduction. The colony grows, and then we're able to passage those pluripotent stem cells. Okay, there's a lot of work to be done before we really convince ourselves that these are, in fact, pluripotent stem cells. We want to know for sure first that genes like TE7 that are expressed in fibroblast cells go away, that they're downregulated with dedifferentiation to iPS cells, and TE7 does go down. We want to know that proteins like NANOG and OCT4, which are expressed in embryonic stem cells and in embryos, are expressed in the cells to convince ourselves that they are, in fact, reprogrammed, and these are pluripotent cells. We want to know that the cells can undergo lineage differentiation to multiple cell types, just like the embryonic stem cell. So they express smooth muscle actin, which is expressed in muscle cells. They express TUJ1 in the nervous system, SOX17 in endoderm. And we confirm this both at the protein level as well as in gene expression studies. Okay? So far we've made stock vials of fibroblasts, so we can always go back and reprogram more cell lines if we need to in the future. We've carried out the immunohistochemistry. We believe they're induced pluripotent-like stem cells. We've looked at gene expression, and we believe that we've now successfully reprogrammed skin cells from people with bipolar disorder and normal control individuals to be pluripotent. So now we can begin to differentiate these cells into neurons and glial cells, and we've taken a pretty standard approach to doing this in tissue culture. After a couple of weeks, we pick and differentiate the cells into primitive neurons. We're particularly interested in looking at gene expression profiles, so we've done microarray analysis on the induced pluripotent stem cells, as well as the neurons that are derived from those stem cells. After about 4 weeks of differentiation, these are the control cells on the top, and the bipolar induced pluripotent stem cells that have differentiated on the bottom panel. And you can see that the control and the bipolar cells now express markers of neural stem cells. They express this protein that's called nestin, another one that's called SOX2, and they're capable of differentiating into early neurons. If we grow them for another couple of weeks in tissue culture, we see clusters and clusters of neuronal cells with long interdigitating processes, both in the control and in induced pluripotent stem cells from bipolar skin samples. They form axons and dendrites, and we're just beginning now to characterize their neurotransmitter phenotype. So we understand a little bit about how the neurotransmitters may be different between these two populations. So I told you earlier that we did microarray analysis to look at gene expression differences between these two populations. We looked at gene expression in induced pluripotent stem cells. We did 6 different lines from 6 different individuals. And in neurons, again, 6 different individuals were analyzed. And we found a really interesting series of genes that were differentially expressed between the two populations. One cluster is involved in development, and particularly development of the nervous system, cell migrations that might be involved in setting up the cortical layers as the brain develops. And a second group of genes that are involved in sequence-specific DNA binding. These are transcription factors that control expression of genes that are involved in differentiation to the various lineages. We went on to stratify the data a little bit more. We looked at genes that were upregulated in bipolar versus control neurons, and every single cluster we found had to do with something involving the plasma membrane and its organization. So we found that in bipolar versus control neurons, calcium channel genes were up, potassium channel genes were up, receptors for neurotransmitters were up, cell-cell interaction molecules were up in these cells compared to the controls. We looked at genes that were downregulated in bipolar versus control. These were all transcription factors, as well as genes that are involved in cell cycle, determining how fast the cells divide in tissue culture. There's some very interesting genes in this group, the ataxins, cyclin genes, again, how often the cells divide. Eomesodermin, which is also TBR2 and TCF4, involved in the development of the forebrain of the early embryo. So we have some really interesting, great candidates to pursue. So overall, our conclusions are we can make these induced pluripotent stem cells from adult tissues, from bipolar and control individuals. They are capable of neuronal differentiation. They form widespread neuronal outgrowth, differentiation of axons and dendrites, and gene expression profiles have identified genes that are involved in CNS development, in organization and signaling via the plasma membrane, and also transcription factor activity. So we have a lot of work to do. One of the things I most want to do is do what's called checkerboard analysis. So we're going to take iPS cells from bipolar, turn those cells into neurons, and co-culture them with glial cells from control lines. And we'll do the reverse. We'll make control neurons and culture them with glial cells that are derived from bipolar individuals so that we can mix and match and figure out if one cell population is differentially affected. Okay? The other things we need to do are characterize the neurotransmitter phenotype of these cells. We particularly need to stimulate these cells with lithium, with small molecules to identify new pathways, druggable targets in these cells. We're carrying out calcium imaging studies in collaboration with Dr. Todd Herron. He's a cardiologist, and of course they're also interested in excitable tissues. We're able to look at calcium flux. We're able to stimulate these cells with agonists and see what the cells express into the tissue culture medium. So we'll be able to look at cytokines, neurotransmitters, chemokines, neuropeptides that are released by the different groups of cells. I'm really very interested in transplanting these neurons into animal models, into embryos, into maybe the hippocampus of animals to see if the cells will integrate, whether they will die, whether they will interact with other cells, signal back and forth to the other cells. And I'm particularly interested to determine if the recipient has any sort of behavioral changes that we might be able to identify after transplantation. So I think I'm out of time. I really want to thank a lot of people today. First of all, our funding sources, particularly the Prechter Bipolar Research Fund, the Schwartzberg Memorial Fund, the study participants, particularly for your time and effort, the staff at the Rachel Upjohn, the Depression Center, the Department of Psychiatry, my colleagues in the Department of Cell and Developmental Biology, the Taubman Institute for supporting us, and the staff of the CSCT, particularly Cindy and Haiming, who have done all the work that I talked about today. If you're particularly interested in stem cell biology and you'd like a little bit more information, there are a couple of really good websites. The first is just stemcells.nih.gov. You can find out about stem cell clinical trials. It gives you a wealth of information about what stem cells are, what cells are available to study. Not to be outdone, the University of Michigan also has a tutorial that's pretty good. It's called umich.edu/stemcells/tutorial. And of course, I'm always willing to take any questions that you might have about stem cells. So again, in the spirit of ethics policy and as a brief introduction to Mr. Goldman here, this is one of my favorite sites. This is our lab, the small little Consortium for Stem Cell Biology lab. And this, of course, is the oversight committees at the University of Michigan. Thank you.